Okay, let's do a little review of the biological macromolecules. I'm going to start with just what are they, and here you can see we can look at four classes of these molecules. Things we put under the umbrella of carbohydrates, or lipids, proteins, or nucleic acids. Now I'm sure the first three you have no problem thinking about them because all of them relate to food. Remember, the nucleic acids are related to things like DNA and RNA. But all of these are organic. All of them are going to contain carbon, which is one of the requirements that when we say something is organic, it means it's a carbon-based compound, and that most of them also contain a fairly limited set of other elements such as hydrogen and oxygen, nitrogen and phosphorus and sulfur. So those are the ones that we typically see in macromolecules. Macromolecules, macro means large, right? So when we look at macromolecules, we're looking at something that has is big, and so it's built though from these things we call monomers. So monomer is just a term that means just the individual unit of the of whatever structure we're talking about, and that they build into things we call polymers. So there are a lot of polymers around us, including things like plastics. So sometimes you'll hear that term in reference to things like plastic, but we're looking at it from the biological standpoint. So we're going to look at some of these groups here in a minute. But the question is then, how do we create these large polymers from individual units or monomers? And that's through this process we know of called dehydration synthesis, where two of the individual units create a bond between them, and part of the activity that's involved with that is the removal of water. So we have one water molecule that is pulled out, part of it coming from one of these monomer units, part from the other, and we can see that it's a product at the end. But then we have a bond that's formed, and this bond is a covalent bond. So it's a strong bond. We can reverse that taking a larger unit. This is obviously a disaccharide here. We have two sugar units here. We can use water in that case, so water then being a reactant is going to break apart this bond, and then the parts of water get added back in. So this is the reverse type of reaction. And this is showing so these are both glucose and glucose, each of these units is glucose, but this is maltose. So this is formed this disaccharide from two glucose monomers. The other thing that we'll talk about, even this really fits more under the umbrella of proteins because enzymes are proteins, but we know that enzymes are really important in all chemical reactions. They're going to help to catalyze or speed up a reaction. And so all of the processes that we talk about in biology, almost without exception, involve enzymes. So enzymes are going to be involved with both hydrolysis and dehydration reactions, but enzymes are very specific to what they break down or what they build up, and so there are enzymes that work specifically with carbohydrates, with lipids and proteins, and here's some examples of that. So you recognize, we always come to this one because it's familiar to us, lactase, being an enzyme that's specific to breaking down the sugar lactose in milk products. And then we have lipases, which break down lipids, so that's just a kind of a group name for those types of lipids. And then we have some more specific ones here for proteins. So now let's just start with carbohydrates. We know that what they are. We see representations here of foods that we associate as being carbohydrates. They're literally hydrated carbon. So here's the carbon and here's the water. So this would be the generic formula for a carbohydrate, the general formula, where you have a ratio of carbon to hydrogen to oxygen of 1 to 2 to 1. The groups that we've talked about, monosaccharides, disaccharides, and polysaccharides. Now, we spent a while talking about different ways we actually classify carbohydrates in particular because we looked at some of their different structures. So we know that they have, the ones that we're most familiar with have between 3 and 7 carbons, many of them having around 5 or 6 carbons, that the suffix OSE at the end tells us that we indeed have carbohydrates, and that all of them have this functional group. This is that carbonyl group, where if it's at the end of the chain, we say it's an aldose form. If it's within the chain, we say it's a ketose form. So those are that's all related just to this carbonyl group. But then we could also use just the counting of how many carbons there are. We can talk about a triose as any of these carbohydrates with 3 carbons. Pentoses have 5, hexoses have 6. So you're going to see those terms and have seen those terms as we were discussing this before. So here are 2 3-carbon monosaccharides, which we don't, here's aldose, and here's a ketose form. Sorry, this is an aldose form, this is a ketose form. We won't see these until later on, but we don't see 3-carbon ones as frequently as we do some of the others. Whoops, kind of got carried away there. So here's glucose, ribose, and this one that was above, glyceraldehydes, is showing a triose, pentose, and a hexose. All of them, though, are aldoses. So if I'm classifying by where that carbonyl group is on the molecule, then I can call all of them, regardless of the number of carbons, aldoses. Here's another example. In this case, though, what we see are things which look almost identical and, frankly, actually are identical in terms of their formula. So all of these have the same C6H12O6 formula, and because they all have that same formula, but there's just some difference in the arrangement of those atoms in that molecule, we call those isomers. And so here we have glucose, galactose, and fructose, all of which are monosaccharides. And with glucose and galactose, we could say they were hexoses. We could also say that fructose is a hexose because it has 6 carbons. But these two, we say, oh, these are aldoses, and this is an aldose, and this is a ketose. In the body, though, in aqueous solutions, which is really us, right? We're a very watery thing. We have water on the outside of our cells. We have water on the inside of our cells. In most of those under those conditions, most of these monosaccharides form these ring structures. So what we see here on the right is this was one of those linear ones that we saw a minute ago. There's glucose, and it's showing what happens when it becomes cyclized or forms these ring structures here. If it goes in one direction, if this links, this is called an anomeric carbon, which you don't need to know, but this can kind of go in either direction. It can rotate. So in the end, what we're going to see on this molecule over here is the OH down here, or is the OH up here. This is the alpha form. This is the beta form. Now that alpha beta might be familiar to you because we are about to talk a little bit about how bonds can form from the alpha or from the beta when we get to polysaccharides. Down below you can see, I'm going to move this over a little bit here so you can see here's the 2 5-carbon structures. So we have a ribose and we have a fructose here. So I'm sorry, not 5. It's not 5 carbons. These are just these different formations. So these came from where this up here was an aldose form. These down here were the ketose forms. So you can see that. All right, so here's how disaccharides form. We have on this side we have a glucose, which you notice is a nice pentagon shape. We have a fructose, which is that, hexagon shape. Here's your pentagon shape. But here's where the water is being pulled out. Dehydration synthesis is forming this thing we call a glycosidic bond. Now here, this is called a 1,2 glycosidic linkage. Remember, those linkages really applied a lot when we talked when we'll talk about polysaccharides, but it's just showing us that it numbers the carbons. So here's the carbon, on this structure, this is carbon number 2, and on this structure it's carbon number 1. So that's why this is a 1 to 2 glycosidic linkage. It's called a linkage, even though it is a bond too, but it's just describing where those monomer units are actually linked together, which carbon. Here's some other common disaccharides. Sucrose is the one that we use the most frequently, at least at home, right? Because this is table sugar, this is the white sugar that is often used in cooking. And then there's lactose, and then there's maltose. So all of these are disaccharides. And you can see how they might link, whether we're getting this type of a formation or this type of a formation. Here we go. So polysaccharides we spent a long time on because these are the ones we come across a lot. And they have many monosaccharide units that are linked again by glycosidic bonds. They can be straight chain or they can be branched. And so we're going to see a couple of them coming up here. So starch is a polysaccharide, and it's made up of these units called amylose and amylopectin. Starch has a couple of different bonds in it, so it forms both 1 to 4 and 1 to 6 alpha glycosidic bonds, and you can see them over here on this picture. These are the ones that are kind of going down. These are alpha linkages, and the reason starch is valuable, or we don't make starch, we get starch from plants. Plants are the ones that produce this. This is a storage form of energy for a plant. And so when we eat it, then we break it down and build other things. So amylose happens to be the unbranched kind of version, and then the amylopectin is the branched version. So you get some of both in this particular. You get this, and you get these branched points. Where cellulose, which this, as I'm looking at it right now, I'm saying, that's misspelled, cellulose, there we go, is found, is what we talk about all the time as being, having beta linkages. And this is really right here where I'm circling. Here we can kind of more clearly see a beta linkage. But the reason why, these are all betas, but some of these monomers are flipped in different directions. So the fact that these are all beta linkages is important in that we can't break these down. We don't have an enzyme to break these linkages. So that's why eating vegetables, a lot of that vegetable material is that cellulose. It's making up the walls of the plant cells, and we can't digest it, we can't break it down, and therefore it's what we call fiber. And that's because we don't have the enzymes to break these beta glycosidic linkages. And then we're about to get into lipids, and I will stop there and start again with a little review on lipids.